Hello student, today we are going to start a new chapter, that is biotechnology and its applications. In the last chapter, in chapter number 11, we have discussed the principle and processes of biotechnology. Now, in this chapter, we will see what are the significance or what are the applications, how can we use biotechnology for the benefit of the mankind. Before going on some of the chapters, some of the applications or the products, okay, or the proteins that have been synthesized, let's try to understand the meaning of transgene. Okay, transgene means, here we are introducing the gene of interest, right? Or we are altering the gene. We are inserting the foreign gene. I'll say that inserting the foreign gene. Foreign gene means the outside gene, okay, that have been extracted from the other source, okay, and then inserting the foreign gene into, and what will happen if you put in a host, okay? The host can be plant, animals, fungi, or it can be bacteria, right? So when you insert the foreign gene to any of the host, what will happen? That will alter the genome of the host. That is called the transgene, okay? Once we introduce the foreign gene, means the gene of interest, okay, depending on our purpose, right? And then we introduce into the host. Then what will happen? The genome of the host will be changed, right? The characters will be changed, okay? And we call that as transgened. And when those transgenic organisms, okay, the organisms, if you put in a plant, we call transgenic plant. If you put in a, if you insert the foreign gene, okay, if you alter the genome of a particular host, okay, it can be plants, it can be animals, it can be fungi, it can be bacteria, okay. And if we manipulate the gene, if certain manipulation have been done with the help of biotechnology, we call that as genetically modified organisms. In simple, we say that gene manipulation, okay. Those organisms where the gene has been manipulated, and we call that as genetically modified organism. Now, what is the significance of these transgenic organisms? Okay. Now, if we take an example of transgenic bacteria, transgenic bacteria. What is the meaning of transgenic bacteria? Means you have manipulated the gene of that bacteria, and then we can use these bacteria as a living factories. Okay, point number one to be noted is living factories. Now you can produce protein, living factories. So from this bacteria you can produce proteins, you can produce enzymes, you can produce drugs, you can produce vaccines, and most important thing is that these transgenic bacteria can be used to degrade waste. Okay, environmental pollution can also be degraded, okay, with the help of transgenic bacteria. Let's see for transgenic plants. Now, when we say plant, it is related with the crop, like increase in the productivity of the crops. Say green revolution, okay, where we need, due to increase of the human population, we need higher yield of the crops. So, with the help of transgenic plants can give us the quality products, okay. Say point number one, like nutritional value, okay, in the plants. Or we can also say like these plants can have abiotic resistance. They have a ability to certain stresses, just like drought, okay. If the soil content salt, more salt condition, more drought, flood, but still our plants will survive. If transgenic plants can survive, it means that our productivity will be increased, right? So if we have certain abiotic stress, you can see during the hot season, you can see many crops where they are not able to grow. okay, due to less sufficient of water, right, or because of the flood. So transgenic, they has the ability, those plants has the ability to resist, okay, has the ability to provide resistance to certain abiotic stresses. And the most important thing is that they can also increase longer life shelf, okay, or the post-harvest, okay, reduce the post-harvest, reduce the post-harvest. So this is very important, okay. Now this is about the transgenic plants. Now in the short, I want to discuss even about transgenic animals. If we manipulate the gene of any animals, okay, with the help of biotechnology, what are the applications? What are the significance of it? Okay, see, milk. Okay, milk products are in need in this 21st century, and then the milk quality or the productivity can also be enhanced. You know, and then fleshed. We need meat products. Okay, the fleshed can also be yield. Okay, and these transgenic animals, okay, we can also use as a in the field of research. Okay, so these are some of the significance of our transgenic organisms. Now we are going to talk. I'm going to discuss very important topic, that is BT cotton. Okay, one of the transgenic plants. Okay, and this BT cotton. Okay, many of the time this question was asked in your, even in your competitive examination and even in your board examinations. Okay, so let's discuss BT cotton. Now this BT cotton, when I say BT cotton, you know what is cotton, right? So this cotton, okay, it means that when we say BT, it is about Bacillus thuringiensis. This is a bacteria, right? Let's try to understand this Bacillus thuringiensis. Okay. Now, usually this microorganism, this bacteria, they kill insects. They has the ability to kill insects. Just say like diphterans, okay, like houseflies, mosquitoes or beetles, okay, they can, like armyworm, bollworm, cottonworm, okay, they can kill those insects, these bacteria. Now how they can kill or how they can fight, okay, is that because they have these Bacillus thuringiensis in their certain group of their life cycle, okay, in a certain group of their phases of life cycle, they produce crystal protein. Crystal protein. This crystal protein produced by this Bacillus thuringiensis. Now this crystal protein, they contain toxic insecticidal protein. So this crystal protein has some toxic insecticidal protein. Okay. Now, but these remain in an inactive form. They are not active, but they remain in an inactive form. Now, once the insect, okay, once the insect ingests these, digest or take up the inactive form, there is a crystal protein. This inactive form turns to toxin or BT toxin active form. Okay, so it happens only when the insect digests. Once the insect digests this inactive form, that is the crystal protein form, then this inactive form will solubilize. Just say that this is our gene, the inactive form. Okay, so they will solubilize this. They will break this. Solubilize. Once this solubilize, they will convert to BT toxin active form. Okay. Now, why? Because in insect, the conversion or the conversion of your inactive form to active form is because of the pH. Okay, pH of the gut. This is alkaline pH. Okay, so this question was asked in your competitive examinations. Okay, so alkaline pH of the gut of the insect. Because of that only, the inactive form get converted to active form. Once this active form converted, means inactive form has been converted to active form. This active form is Bt toxin. Okay, it will go and bind to the surface of epithelial cells. This is just the surface of the midgut of your epithelial cells. Okay, midgut of the epithelial cells. Very important point to remember. Okay, now once this toxin binds to the surface of, just say this is your midgut of the insect, here the Bt toxin will bind. Once it binds, what will happen? This will create pores. Okay, they will create pores. Okay, they will make holes on the surfaces of these epithelial cells. Once it creates the pores, this will result to swelling and lysis. And ultimately, what will happen? The insect will die. Okay, the insect will kill by bursting the stomach of that insect. Okay, so this is a mechanism that had been observed in the Bt in Bacillus thuringiensis. So biotechnology, now we are engineering. Okay, we try to search the gene of that Bt toxin. We know that the gene is responsible for creating or for producing this useful toxin as a defense mechanism. So this Bt toxin, researchers, they found that why don't we use this Bacillus thuringiensis gene, and that is known as Bt toxin gene, and that is Cryogen. In simple, we call Cryogen. Okay, so for cotton bollworm, just say Cry1 AC, okay, Cry2 AB. Okay, Cry2 AB. This is for cotton bollworm. Okay, likewise we have Cry1 AB. Okay, this is for corn borer. Okay, corn borer. So if we introduce, okay, the Cryogen was introduced in a cotton. There is a cotton bollworm. So in a cotton plant, if you introduce this Cryogen, in a cotton, it means that Bt cotton is a genetically modified plant. Okay, you have introduced a gene that will fight against the insect that destroy the cotton plant. Just imagine that you have grown a cotton farm, and then for year to year you have been looking up, and suddenly the yield is not good because of those insects that destroy your cotton plant, right? So in order to produce high yield of cotton, okay, the uses of biotechnology is very important. You can introduce a Cryogen, okay, in a cotton, and then we can produce a genetically modified cotton plants. So if we introduce this gene in a cotton, what happened? Even insect comes and try to eat the leaf, then the Cryogen will get activated in the stomach of that insect. It will produce crystal protein. This crystal protein will solubilize to convert to inactive form to active form. this active form now result to Bt toxin, which will destroy the insect. So this is a simple mechanism, okay, or the simple applications, okay, how to use microorganism, okay, for the beneficial of the mankind. In the next class, we'll talk about the RNA interference or pest-resistant plant and human insulin, that is genetically engineered human insulin. Thank you.